Davison. I'll be talking today about phenotype-first evolution of cellular differentiation in the volvocine green algae. How do complex novel traits, like new cell types, originate? Now, most of you are probably familiar with the gene's first pathway, which occurs when genetic changes lead to corresponding phenotypic changes. But the phenotype-first pathway is much less well understood, and it occurs when a trait that's initially expressed plastically becomes secondarily fixed and modified by adaptive evolution. Now, it's unclear if this pathway can underpin the origin and evolution of a trait like a new cell type, which is an important evolutionary innovation. So here, I'm asking if the plastic production of a new cell type preceded its fixation via genetic assimilation. And I'll be using the volvocine green algae as a model to address this question. The volvocine green algae are a particularly good model system for studying the evolution of cellular differentiation. Because you have some species, like this one here, the Pseudorhina, where all of the cells are identical, undifferentiated cells within one simple multicellular organism. But then you have closely related species that have somatic cells, which you see here. See those small little somatic cells? Those do not reproduce. And in other species, you see thousands of somatic cells. Those are all the little dots in there. Now, cellular differentiation has been well characterized in this clade. There have been multiple gains and or losses of soma within the volvocine green algae. And you see that by looking at the phylogeny here. So all the species shown in black there are species that are soma differentiated, so they have somatic cells. And all of the species shown in gray are species that are characterized as always being undifferentiated. The genetic basis of somatic cells has also been characterized in this clade. We know a group of genes called the Reg cluster may be important for somatic cell developments. And I'll be focusing today on the Eutorhina clade, which is shown here in that green box. So if you just look within that Eutorhina clade, you see that there have been multiple gains and or losses of soma. And you see species that have no cellular differentiation, like this one here, and species that have somatic cells as well. So I'm hypothesizing that somatic cell development was plastic in the undifferentiated ancestor of the Eutorhina clade. If this was the case, I would expect that undifferentiated Eutorhina species would develop cells that resemble somatic cells in response to environmental stress. So these cells should be small, they should have flagella, they should not reproduce, and they should be living cells. They should also be plastic and not the result of a mutation. I also am hypothesizing that the development of soma came under genetic control in some lineages via genetic assimilation. And if this was the case, I would expect the ancestral state of the Eutorhina clade to be plastic soma, and that somatic cell development would be under developmental genetic control in some species. So to test this, I'm focusing on two different Eutorhina elegans species, Eutorhina elegans Utex 1201 and Nis 457. They have the same name, but they are phylogenetically distinct. They're two different species. And they're both located within this clade, smaller subclade here, which is an outgroup to the rest of the Eutorhina clade, and most likely never had an ancestor with somatic cells. And I'm taking these two species and I'm subjecting them to an acute environmental stressor. It's 2 hours of cold shock, and then I'm imaging them. And when I did that, I found initially that the Eutorhina elegans species that normally have no somatic cells, they're usually undifferentiated, I saw a small number of smaller cells that, on first glance, resemble soma. But are these actually somatic-like? Do they have flagella? Are they alive? Do they fail to reproduce? Are they actually smaller consistently smaller than reproductive cells? Additionally, is their development in fact plastic, or is it the result of a recurrent mutation induced by cold shock? We know that transposable element movement can be induced by cold shock in related species, so this is plausible. So first, I cold shock these colonies and I stain them with a stain called fluorescein diacetate, which permeates cells, and if those cells are alive, it's cleaved by non-specific enzymatic activity in those cells fluoresce. So you see here that the large reproductive cells are fluorescing, like you would expect, but these small somatic-like cells are also fluorescing, because they are most likely alive. Additionally, I imaged these, took a video of these colonies, and you can see the somatic-like cell right here has flagella, and you can see that those flagella are beating. I also found that these somatic-like cells are in fact consistently smaller than the reproductive cells. Here you see the somatic-like cell diameters for UTEX 1201 compared to the reproductive cell diameters for that same species. Same thing with MES 457. The somatic-like cells are smaller. Additionally, they do not reproduce. And you see that here. Here's a colony with one reproductive cell, 7 somatic-like cells. That reproductive cell divides, the daughter hatches out and swims around, and the somatic-like cells remain in that extracellular matrix. They do not divide. So I showed you that these cells are similar to soma. Now I need to determine if they are in fact developing plastically. And to do this, I had to track individual colonies and their descendants. So I took single colonies out of a flask, and I transferred single colonies into individual wells and well plates. And then I imaged the colonies that I cold shocked directly. I imaged their offspring, and I also imaged their grand offspring. And I did this cold shock by placing colonies in Eppendorf tubes in ice water. And I found that when colonies are cold shocked directly, they do develop somatic-like cells. So here you see the proportion of colonies that have somatic-like cells for the control. All the ones in light green are colonies that have one or more somatic-like cells. You see it in the control. There's a small proportion of colonies that have those cells. But in the cold shock treatments, all of the colonies developed one or more somatic-like cells following cold shock. I then looked at the offspring of those cold shock colonies. And again, here you see in the controls, a small proportion of colonies have somatic-like cells, most do not. But in that cold shock treatment, about 50% of the offspring of my cold shock colonies had one or more somatic-like cells, which means that colonies with somatic-like cells give rise to daughters with those cells and daughters without them. I then looked at the grand offspring of those cold shock and control colonies. For the controls, I compared the generation 3 colonies whose parents had somatic-like cells to those that lacked somatic-like cells. And I saw no difference there. Same thing with the cold shock colonies. Again, the proportion of colonies that have somatic-like cells is independent of whether or not their parent had somatic-like cells. So this trait is most likely not the result of a recurrent mutation. Now that I've characterized these somatic-like cells, I need to determine if the somatic-like cell development is ancestral to the uterine clade. Thus far, I've just focused on species that fall into this smaller group that's the outgroup to the rest of the uterine clade. So it's possible this could just be a trait that's specific to that lineage. Alternatively, it's possible that the somatic-like cell phenotype may have arisen somewhere back here, and it may have even arisen the same time as the genetic basis of soma. So to start examining this, I cold shocked additional Uterina elegans species, that fall in a completely different portion of the uterine clade right here. And I found that somatic-like cells do develop in these uterine species days after cold shock. You see this is true of my first uterine species. You see that there's a higher proportion of colonies with cold shock, with somatic-like cells in the cold shock treatment relative to control. And this is consistent with what I saw in the second uterine species. So the ancestor of this uterine clade most likely had somatic-like cells. So somatic-like cell phenotype is going to be, it's most likely ancestral. Now, all the species that are shown in black here have previously been characterized as always having soma. So this ancestral plasticity would have been lost in these species that always have cellular differentiation. In other words, somatic cells may have evolved via genetic assimilation in this clay. So let's return back to this initial question. Did the plastic development of a new cell type precede its fixation via genetic assimilation? So today, I showed you that the development of somatic-like cells was most likely plastic in the undifferentiated ancestor of the uterine clade. These undifferentiated species develop cells that do resemble soma in response to cold shock, and the development of these cells was plastic, not the result of a recurrent mutation. I also showed you that somatic cell development is under genetic control in some of these lineages. Numerous soma-differentiated species have been characterized in this clade, and the genetic basis of soma has even been characterized for this lineage. So I'm currently looking at the genetic basis of somatic-like cells. I'm also examining the phylogenetic distribution of this phenotype, looking across the volvocine algae phylogeny. So today, I've told you a bit about a novel framework for understanding the origin of new cell types using the volvocine green algae as a model system. I showed you that somatic cells in these algae may have originated as a plastic response to the environment that later came under developmental genetic control, or later refined by adaptive evolution. I'm currently examining the phylogenetic distribution of the somatic-like cell phenotype, and I can then compare this to the origin of the genes involved in cellular differentiation in this clade. Additionally, I'm examining the molecular mechanisms underpinning somatic-like cell development, and I hope to talk to you more about these last two at a future evolution conference. So I'd like to thank all of my collaborators and my funding sources and all of you for attending. Thank you. Yes.
So why cold shock?
So that's a great question. So I actually initially chose cold shock as part of a different experiment entirely, where I happened to observe this phenotype as a result. I'm right now running experiments to determine if this is just a cold shock specific response. The short answer is that it does not appear to be. The being grown at high densities with low nutrients also appears to induce the development of these somatic-like cells. But the cold shock is an acute stressor that does so consistently. Yeah. I guess I'll take one more question while the next talk is being set up. Yeah.
In other volvocine species that have soma, it's usually spatially organized. It's either in the anterior or the posterior.
Yeah.
Do you see any evidence of that?
I'm currently looking at that. They don't appear to just be randomly spread out around the colony. They tend to be clustered when there's multiple somatic-like cells. Great question.